Okay, let's dive in. Imagine using AI not just to, analyze things, but to actually create or simulate incredibly complex stuff. We're talking biological structures here.
Yeah, it's a fascinating shift, isn't it? Moving AI from just understanding what's already there to potentially generating what could be there based on, well, learning the biological rules from data.
Exactly. So today we're digging into a pretty detailed consultation record. It explores how these cutting-edge generative AI models, things like diffusion probabilistic models and also flow matching, how they're being looked at for generating and reconstructing 3D structures.
And there's a big focus on molecules and protein structures
Right.
specifically. And this record, it lays out the main research goals, the kind of data they think they need, and crucially, some of the, let's say, significant technical hurdles they're up against.
So our mission here is really to unpack those notes for you, give you the core ideas, the big challenges they flagged, and, the potential implications they discussed, get you the essential insights.
Okay, so at the heart of it, the goal discussed in this consultation is developing models that can generate either like discrete things, think individual atoms in a molecule, or continuous representation.
Continue this life.
Like the actual spatial coordinates, the bond angles, basically the stuff that defines the 3D shape. They specifically mentioned using flow matching techniques to generate these 3D layouts.
Ah, okay.
And they note this approach might help bridge that gap, between the distinct atomic points and the smooth, continuous space they actually occupy.
So the AI needs to learn how to go from just knowing which atoms are involved, the discrete part, to figuring out their exact 3D arrangement and connections, the continuous part.
Precisely. And the data sources mentioned in the record for training these models, they reflect this need. You've got small molecule databases like QMO9 or Zinc25K.
Right, libraries of known chemicals.
Exactly. And then also protein structure data, maybe coming from things like AlphaFold predictions, which gives insight into how those amino acid chains fold up into complex shapes.
AlphaFold, yeah, that was a huge step in predicting protein structures.
It was. And that leads right into one of the major challenges they specifically call out in these notes. Proteins, especially, follow incredibly strict physical and chemical rules.
Like bond geometry, how atoms interact, the overall folded structure.
Exactly. And the consultation really emphasizes that these AI models have to somehow incorporate this fundamental biological knowledge, this prior information.
Otherwise, the AI would just be spinning its wheels, computationally speaking, trying to relearn basic chemistry and physics, and maybe generating junk structures.
That's the worry, yeah.
Yeah.
The advice in the record is pretty pragmatic. Use what we already know about biology. Start with known structural patterns, conserved evolutionary motifs, maybe templates.
Kind of like how the best protein prediction methods don't start totally from scratch.
Precisely. They build on existing knowledge. The note suggests this knowledge injection is, well, critical for generating biologically valid and stable structures.
That makes a lot of sense. Build on the science we have. Did the record offer any other sort of strategic advice for handling this complexity?
It did. One key recommendation was to start by training models on data representing normal biological states,
Yeah.
healthy structures or processes.
Before trying to simulate disease states.
Right. Get a solid baseline first.
That seems like a logical way to progress.
It is. And they also talk about applying this generative idea beyond just static structures. The record discusses using these methods for looking at dynamic single-cell RNA sequencing data.
So tracking changes over time.
Exactly. Understanding processes like, say, cellular differentiation flows, how a cell changes state, or predicting cell fate. They frame this as fundamentally a generation problem.
Because you're modeling the process, the trajectory of change, not just identifying a cell at one snapshot in time.
Correct. And single-cell data has its own issues, like sparsity, lots of missing information. So generative models are proposed there, too, for filling in those gaps. Imputation, they call it. And a practical tip mentioned in the notes was maybe filtering out housekeeping genes.
The basic survival genes.
Yeah, the ones active in almost all cells. Filtering them helps isolate the gene expression patterns that are really specific to the differentiation pathway you're trying to model.
Makes sense. And how do they suggest evaluating if these generative models are actually working well? What are the success criteria?
They strongly advise using those fundamental biological, physical, and chemical rules as the main evaluation metrics.
So does the generated molecule or protein actually obey the laws of chemistry? Is it structurally stable?
Exactly. That's seen as a crucial check for whether you've generated something reasonable or, biologically plausible. Ultimately, the record recommends starting simpler.
like with small, well-understood molecules.
Right. Build confidence, verify the models, work on simpler cases before tackling the huge complexity of large proteins or dynamic cell systems. They even suggest developing specific analytical tools alongside the models to help researchers actually explore the generated cell states or track simulated gene expression.
So pulling it together from this consultation record, it's clear researchers are really pushing generative AI to simulate life's building blocks and processes. It's moving beyond just analysis.
To actually generating what might be possible, from small molecules up through protein structures and even mapping how cells change over time.
And understanding these methods, these challenges they've outlined, it seems really valuable. Whether you're in AI or biology, these techniques could lead to some pretty big breakthroughs.
Oh, absolutely. Things like designing new drugs, creating much better models of diseases, or just, fundamentally getting a deeper handle on how biological systems work at that intricate level.
It really does make you think. If AI can truly learn and reproduce the complex physics and chemistry that govern biological structures, basically, the logic of how life is built, what does that mean for our future ability? Not just to understand life, but maybe to actively design completely new biological entities or simulate complex life processes entirely outside a wet lab? It's a pretty profound question.